Today we're going to talk about an interesting and unique group of organisms in freshwaters. These are the pearly mussels, and they're a group of freshwater mussels that I think are really interesting and are uniquely diverse in North America. So we're going to first cover basically how they're named and how they relate to other named groups of organisms. We're going to talk about the diversity and distribution of the mussels, of the freshwater pearly mussels, a little bit about how they interact with other elements of the aquatic ecosystem, as well as how they played a role in human history. We'll talk about their life history and adaptations, and then we'll compare those adaptations with other groups of freshwater bivalves. So, they're in the phylum Mollusca. They share that phylum with other organisms like the octopus, the squid, and even snails. They're in the class Bivalvia. These are organisms that have two shells on either side of their body, the left and the right sides. They're in the subclass Paleoheterodonta and in the order Unionidae. We're going to focus on a group of families within the Unionidae that have a unique life cycle. And we'll get to that in a little bit. In the order, there are about 900 species, and we're going to focus on two families that are present in North America and Europe, the Unionidae and the Margiferidae. Mostly the Unionidae, that's where most of the North American species come from. So to put some of this taxonomy into perspective, both the Dracenids, a common invasive species, a common invasive family in North America that's well known because of effective public outreach campaigns and really tremendous ecological and economic damage that they've caused, are about as related to the Unionids in terms of just naming conventions. So this doesn't take into account anything like genetic distance or evolutionary distance, things like that. But by naming conventions, the Unionids and the Dracenids have about the same level of name similarity as we do with lemurs. Just let that sink in. Because if you know the zebra and quagga mussels, you probably don't know the Unionids. They're very different. I also want to make the point that they're not the same as the mussels that you normally would get to eat in a restaurant. Those are a whole other group that come from marine systems. They're the mytilids. And a lot of these are blue mussels. And they, too, are very different. So when we look across North America and the rest of the world, we've got about 300 species in North America. About 900 species of these pearly mussels in the world. And as I go through some of these pieces, we find pearly mussels everywhere in the world except Antarctica. Big surprise. We can group them into a superfamily, the Unioidea, and 6 families within that group. Again, varied estimates. Around 850 species globally. In North America, we have around 300 species. Again, I think some of this taxonomy is changing, is going to be changing, especially because we're starting to combine traditional morphological, looking at the shapes of these organisms, at the anatomy of these organisms, and we're combining that with the genetics that sometimes tell a different story. And I think that's going to be an interesting thing to follow as we work on the conservation of this group. In Wisconsin, there are roughly 51 species, and overall, roughly 70% of these species are threatened or endangered or are in some other way in need of conservation. And really in imminent need of conservation. And again, we'll come back to that point throughout the lecture. So if we wanted to look at how these organisms relate to the other families that I mentioned. This is a great phylogeny that I've used a couple of times, that I've taken from Dan Graff's work. And you can really see some of the important groups. The groups in blue are the ones that I'm mostly talking about today. The purple line is the Margaritiferids, and these all form generally a clade together. And then we've got these other organisms listed below in these other related families that are not quite as close. So, what do mussels do? How do they interact with their environment, with the other organisms that they share that environment with? Mussels move around in that environment. The pearly mussels are a lot more mobile than a lot of the mussels that we think of in other cases. The dracenids, the middleids, they have byssal threads that attach them to substrates for long periods of time. They're stuck there for a lot of their life. The unionids, they also produce byssal threads, but those are relatively restricted to small portions of their life cycle, and the unionids get up and move around a whole lot more. And part of that, the result of that, is that they interact with the environment and with other organisms in that environment, and can actually increase the diversity and density of other organisms, for example, aquatic insects that share their environment in streams and rivers. So this is a picture of a mussel, one of the Lampsilus mussels, and she's got her foot out and is about to move. She's also got a lure that I'm going to come back to, and you can just barely see it here. And then this is another Lampsiline mussel that's burrowed into the sediment, and all you can see are the siphons. So these organisms filter feed. They suck water through their bodies, take out particles, feed on those particles, and then release the water back into the water column. We've interacted with mussels for a very long time. Humans have used their shells. There's evidence that people have eaten freshwater mussels. One of the most common questions I get during my fieldwork is, can you eat them? And you can. A lot of us who work on mussel conservation would rather you don't. There are a lot of species that are in trouble. It's easy to get the wrong species. That's not always been the case, and people have had a relationship with them. I've heard they're tough to eat, so keep that in mind. But we've used the shells for a whole variety of cultural objects. That includes tools, and then around the turn of the 1800s to the 1900s, we started to see lots of buttons being produced with the pearly mussels, partially because there was an abundance of these mussels in the United States. And later on, it was realized that we can take those mussels and we can produce pearls using the shells. And the freshwater mussels are especially useful because you can take a chunk of them and put them in other bivalves and produce a pearl that's cultured, that's intentionally developed, and that has characteristics that people are looking for. So, rewinding back again to the early 1900s, the pearl button industry, the harvest of these mussels, was incredible. There are stories about being able to walk across major rivers, large rivers, by walking on nothing but mussel shells. And people took advantage of that resource, removed it. These are some historical images that I was able to get a hold of through Illinois, and these images show just the scale of this industry. In a little bit more personal view, these are some photos that I took in Kentucky just outside of Paducah. Here's one from the air, and then here's on the ground. And you can see these mussel shells almost a century after this production had happened, and you can still see evidence of the facility that was here processing those mussels. And so these shells are all the ones that have been drilled out, and then they figured they'd gotten all the buttons out of them that they could, and these are what's left. So one of the things that I think is the most exciting and interesting about these mussels is that they don't reproduce on their own. They reproduce with the help of some organisms that they parasitize. So female mussels take up sperm that's been released and organized into these little balls called spermatozoomata in the water column. They take those sperm balls up, they fertilize their eggs, develop their offspring, and then their offspring have this characteristic larval stage called glochidia, or glochidium, in the singular. Those glochidia almost universally have to attach to a fish and to parasitize that fish before they can grow into a juvenile, undergo a transformation on the fish, and drop off. One of the biggest benefits to the mussel that they get, we know that there's a nutritive relationship, but there's also the ability to move to new parts of that habitat. How do they do that? Well, they mimic fishes. So the mother mussels develop a characteristic lure that looks like a fish. That elicits an attack, that gets a predator to attack that lure, and she then releases her infective larvae, and those larvae then attach to that fish. And I really want to thank Chris Barnhart, formerly at Southwest Missouri State University, for graciously sharing a lot of these images. I've tried to use as many of my own as I could, but a lot of the images in here also come from his work as well. And this is one of those. There are 3 fish and 1 mussel in this picture. Well, there are 2 mussels, but 1 mussel has a lure. And this is what that mussel looks like when she's actively moving her lure around. And you can see some gray lines inside that lure. That's what the fish would bite into, would release glochidia, and that would release glochidia onto the fish. Those are actually parts of her gills. So she's brooding her larvae, these glochidia, in her gills, and then is releasing from the gills onto the fish. Here's another example of a mantle lure. So the mantle is the part inside the shell that's growing out and creating the lure to attract fish. Here's another example of a mantle lure, and this is a female displaying to try and get the attention of fishes. This is a unique species that's actually not imitating a fish, but is imitating a crayfish instead, so a crustacean. And I think it's really important for us to keep in mind that these mussels have never seen a fish. They're not directing this growth consciously. This is something that's happening by adaptation, and is really fascinating to think about, since they can't intentionally grow these things. To the best of our knowledge, can't grow these things in any way that they can mimic stuff that they've experienced. And that's the same, not just with the physical structure, with the morphology, but also with the behavior. So this mussel has turned on her side. That's not a normal position for a lot of live mussels. She's turned on her side and is using that lure to imitate the behavior of the crayfish. But not all mussels create mantle lures. There are other ways that they encourage attacks by fishes. Some of them create structures outside of their bodies that contain the glochidia. In this example, these are conglutinates that are in structures that might increase the likelihood that a fish will attack and be infested with those mussels. And so this is a microscope image of the glochidia packed into that structure before they were released from it. Here's another set of examples of conglutinates. These apparently imitate blackfly larvae. And likewise, when they break open, it releases the potentially infected glochidia. Here's another example of a conglutinate outside the body. And then there are a whole other set of ways that these organisms can encourage infestation on fishes. So they can create a series of nets and traps. And basically these are little mucus nets that are suspended in the water column with glochidia hanging off. Those glochidia can then attach to the fishes as they swim by and as they come into incidental contact with them. There's also a whole set, and I would encourage you to go have a look at these organisms. You can Google epioblasma, epioblasma, and look at the head trapping behavior that these organisms do. So there are some mussels that will actually take advantage of the way that fishes behave on the bottom of the streams, looking for food, and as the fish investigate the mussels, they nose those mussels and clamp onto those mussels and then pump their glochidia over their faces, or over the fish's faces, and that's how the glochidia infest the fish. There are a whole group, a whole array of different morphologies, different shapes. A lot of the ways that we can tell the difference between mussels is by their shape. The color is a little bit more variable. And also, you'll notice that if you go out and look at these in the wild, that the shells they range from really heavy, really thick, to really, really thin. So there are a whole variety of things that we can look for, but I want to encourage you to think about that as the structures. There are bumps, there are ridges, there are what we call a sulcus, sort of a valley in the shell, and all those are unique things to look for. But in a lot of cases, the color doesn't tell us as much. There's some striping that tells us some. And then I want to contrast that with some invasive species, like the Asian clam and the zebra mussel. So if you have a look here, you can see some obvious differences. Asian clams are going to be much smaller than most of our native mussel species. They have really strong ridges. These are the zebra mussels. They have a characteristic flat line along the hinge, and that's where they attach to other objects. Here are some other examples of the dracenids, just so you can have a sense of how they're different in obvious structure from the unionics. One other important picture, and this is a generic veliger larvae that's pictured here, but the dracenids reproduce with veliger larvae. Those larvae can swim, but they're subject to the movement of the water. So they're going to move downstream. And as a result, they move in very different ways. They don't need a fish host, but at the same time, it's much harder for them to colonize new areas through that larval stage. At least under natural conditions. So make sure that you're thinking about that. All right. So what did we talk about? I told you a little bit about the taxonomy of unionid mussels, compared that to the taxonomy, the naming structure of some other mussels that you might be familiar with. And so you should be able to identify what taxa unionid mussels belong to. You should be able to point to where mussels have diversity, and I want to reemphasize that North America has about a third of the global diversity of freshwater mussels that we know about. And that may change as we understand different mussel communities globally. But right now, North America is a great place to study freshwater mussels. We have a lot of diversity here. You should also be able to describe how mussels have been used by people and how they interact with other elements of their aquatic ecosystems. You should be able to describe their special adaptations for getting those glochidia onto fishes. And although I didn't tell you this directly, you should be able to compare that veliger larvae, which is free swimming but also subject to the movement of water, to the glochidia larvae that are parasitic and that are subject to the movement of fishes. And you should be able to make some predictions about how those life histories are going to play out differently. Thanks for listening. I hope you've been interested in this as much as I have, and I appreciate it.